Good afternoon, everyone. Thank you for giving me the opportunity to talk on my research. Today, I'm going to talk on how the segmental duplication landscape can influence uh, the phylogenetic trajectory of species in vertebrates. Uh, Evolvability is defined as a characteristics that describes the capability of a species to respond to selection, and this can be influenced by different genomic structural properties. For example, duplication. Segmental duplication is one kind of duplication that produces genetic redundancy, and that also gives novel functionality, and it also shows elevated mutation rate that produces the potential adaptation. Uh, uh, this figure and the literature that produces this figure shows that the variation in the segmental duplication landscape across across phylogenetic trajectories has a variation, but this variation, but the relation with this segmental duplication landscape variation and the uh, phylogeny is not linear. So to understand the evolvability, I mean how the segmental duplication landscape can influence the phylogenetic trajectories is important. And in the existing literature, the network science tools has an indispensable way to understand complex biological systems, but specifically in terms of the segmental duplication analysis, the study of the network models is very sparse. Uh, Let me show a couple of examples how the network model has been used to understand the segmental duplication particularly. Um, this is one of the representative paper that the first time they proposed network model to understand the segmental duplications. So the construct networks based on the segmental duplication data from Homo sapiens human, where the nodes represents the segments of chromosomes, and the edges describes how two different duplicated segments are aligned. And they showed that this is an example of a segmental duplication network, that you can see that how the fragmented the network is. They found that the size, the distribution of the sizes of this component from the segmental duplication networks is somehow related with the phylogeny of a few number of species. But here, one of the important note is that these species are far away in the phylogenetic tree. So it's kind of obvious that the phylogenetic signals for this species has high level of dissimilarity that can be incorporated in the segmental duplication networks. They also, from the same author, this paper describes that how the genomic feature, for example, the chromatin accessibility recombination rates, this can influence the duplication rate that incorporated in each node in the segmental duplication data. From here, we can hypothesize that the structural properties, the topological properties of segmental duplication networks may have a relation with the evolutionary trajectory of a species. So we aim to provide a empirical network-based analysis of segmental duplication networks for multiple species to understand how the structural properties of these segmental duplication networks can be related with the evolvability. So we constructed a network for 118 species, where the nodes represents the duplicons, and the relation between nodes again describes the similarity, alignment similarity. And indeed, we tested 3 different hypotheses to understand the evolvability of this species. The first hypothesis describes how the segmental duplication landscape varies across species. So if this hypothesis holds, then we can conclude that, that the segmental duplication landscapes is conserved across species. The second hypothesis describes that how the segmental duplication landscape of the species is related with phylogeny. As the species diverse from their recent ancestor, so it is kind of intuitive that that the segmented duplication landscapes also will diverse along with the phylogenetic drift. And the third hypothesis describes, this is basically the alternative hypothesis of the second one, that describes that if the relation between the phylogeny and the segmental duplication landscapes are not strong, then we may conclude that the dynamics of the segmental duplication is very species specific. So as we said, we constructed network, the segmental duplication networks, as we do not have the gene annotation for all of the 118 species. So we make a uniform way to construct networks. So we consider 5 kilo base pairs as a node in the chromosomes. So we have, like, a segment of a chromosome can be duplicated in the same chromosomes or in different chromosomes. And we do have the similarity alignment. So based on the similarity alignment, we can compute the network age weights. And here's an example of a part of networks from Homo sapiens. And here you see that the degree distribution of 3 representative networks, which follows long tail distribution, almost power law distribution, which is very common in biological networks. And to test the hypothesis and to construct the segmental duplication profile, we used different measures related to networks and the similarity duplication scores. And to test the hypothesis, to test the first hypothesis, we consider a threshold on the variation measures 10%. But in our analysis, we found that for none of the 14 measures has the variation less than 10%. Even in some cases, specifically for 6 measures, we do have the variation more than 100%. And in the case of second hypothesis, we follow a guideline with a correlation value 0.3 as a threshold. And we found that for none of the 14 network measures and similarity score, there's no strong correlation with the phylogeny and the segmental duplication landscape. Even the species which has like the divergence less than 50 million years, so they also doesn't show the high correlations with the segmental duplication landscapes. So we conclude that the segmental duplication profile evolved very fast, and the phylogenetic signal becomes very weak compared to the segmental duplication landscape. Here is an example for the clustering coefficient measures that shows that the variation is 28% across the species, and the correlation is 0.04, which is very weak actually. And but interestingly, we found an exceptional case in the case of mammal, where we found that the similarity score of the segmental duplication landscape is very high with the phylogeny, but a more deep analysis shows that this comes from particularly a pair of species, that is monotrim, egg-laying species. And more investigation showed that as the similarity score comes from mismatch score and the gap score, as the dissimilarity score for these 2 particular species is very high, so it's expected that mismatch score and the gap score will have the similar pattern. But we found that the gap score has high values, but not the mismatch score. And of course, the gap score comes from a insertion deletion procedure. So we found that this two particular species went through a distinct genomic structure that derived by insertion and deletion procedure. And as a byproduct, we also try to analyze how the segmental duplications is related with the genome size, as the remnant of whole genome duplications and the localized segmental duplication may have influence in the in driving the genome size across a species. So we try to correlate with the number of duplication and the size of the duplications with the genome size, but in a boring way. But we found no strong correlations, except in the case of fish, where we see that a high correlation 0.53 with the genome size, but that comes from particular species, estrellet, sturgeon, and brown trout that went multiple rounds of whole genome duplications that describes this large genome size. So in conclusion, we say that the past evolving species-specific dynamics dominate the segmental duplication landscape profiles and overshadowing the phylogenetic signals. And we found a particular species in mammals, monotremes, that shows high correlation, but that comes from insertion deletion procedures. And we also found no strong correlation with the segmental duplication landscapes and the genome size. And we showed that the network-based approach can even unravel the complex structure in the genomes. And our data is publicly available, so this may be helpful to understand, to analyze cross-species duplication things. And thank you so much. Our paper is in bioRxiv. If you would like to to read more, then you can go there. And thank you. Thank you for listening to me, and thanks to my collaborators, my advisors, and thanks funding agencies. Thank you so much.
Any questions? Let's give him a round of applause. Oh, sorry.
Yeah.
So are any of your segmental duplications tandem duplications?
Tandem duplication?
Yeah, are any of them next to each other?
Yeah, it's likely.
Yeah. So gene conversion, there's a repair mechanism that can overwrite. If you have very similar copies, one will overwrite the other during the repair, and then you'll get higher similarity than expected. And so I wondered, is that something you looked for? And if you didn't, maybe that's something to look at with your monotremes.
I see. That's an interesting point, actually. Like, I mean, we didn't went through that issues actually, but this is an interesting point. I'll keep in mind. Actually, yeah, I'll do it.
It's worth a, it's worth a look.
Thank you so much.
Give him